I am Annu Yadav from the group number 316. I am here to explain you about my topic, that is protein globule and the hydrophobic interactions. Proteins are the building blocks of our life. It is very essential to have protein in our daily basic diet. Every cell in the human body contains protein. Basic structure of protein is made up of the basic chains of amino acids. You need protein in your diet to repair your body cells and to help your body repair. Protein is essential for the growth and development in the children's, teens, and the pregnant women. A protein globule is the basic unit of the globule that is functional and refers to a compact and roughly spherical structures that the protein assume. And it is a three-dimensional structure. It is folded in many fold by the many bonds like hydrogen bonds, ionic interactions, and many other bonds we will see further. To minimize the protein folds are formed to minimize the free energy in the system. The hydrophobic interactions are the key driving force in the folding of the proteins into the globular shapes. Due to the hydrophobic interactions between the nonpolar solvent and the polar solvent, they occur between nonpolar hydrophobic amino acid residues which tend to avoid contact with water, and then they formed in the inner sphere. In the aqueous environment, these hydrophobic residues cluster together inside of the protein and away from the water due to the hydrophobic interaction. While the polar, there is hydrophilic part. So we see that hydrophilic part is outer and hydrophobic part is inner, that is formed in the folded in the protein. Okay, we will study more about the protein formation. That is, first it is the compact structure is formed. Second, the hydrophobic core and the hydrophilic surface is formed by in the protein. The domains and the motifs like alpha helix and the beta sheets and the turns that it takes, and the ionic interactions and the disulfide bonds in the protein. We see that the protein is also flexible and dynamic in nature, that it is distorted, and it is formed into different shapes. Protein folding and the globular protein examples are like hemoglobin, myoglobin, and enzymes like lysozymes, trypsins, which catalyze the biochemical reaction. Hemoglobin is our multi protein compound that is formed by the four uh, globular proteins, that is alpha and the beta. Okay, two alpha and two betas. The next we will see the misfolding, and the misfolding occurs in the protein is very dangerous for our health, that cause disease like Alzheimer, Parkinson's, cystic fibrosis. In Alzheimer disease, our the amyloid plaque formed in the brain, that is formed by the beta peptides misfolding. Parkinson's disease, it is formed by the alpha synuclein misfolding. Cystic fibrosis is caused by the defective CFTR protein, and we will see the protein stability. Okay, the protein represent a crucial aspects of protein biology, where the compact and intricate folding of a protein dedicates the stability and the ability to protein to perform. Okay, the protein has quaternary structure. Okay, the protein molecule has super molecular protein is system, that is, it contains many geometrical properties of the structures and the forces of interaction and their elements by the interaction with the environment. In protein, when the globular structure undergoes denaturation, it is disordered, and it can forms into many structures. First, if the primary is formed, second the secondary is formed, then the tertiary is formed. It is functional, and quaternary is the multi-unit protein. A typical globular protein is segregation of the hydrophobic and the hydrophilic residues, uh, and this is formed to minimize the free energy of the substance. Okay, Despite the compactness, protein globules are flexible and undergo conformational changes into the different temperature, pH, different environment, and due to the our biological environment. Proteins are, here you can see that proteins are enzymes. Enzymes are the proteins. Sorry, and it catalyze enzyme catalyze are most of the biological environment. So to have protein in our diet is very very essential for to make enzymes and to form the catalysis, to form the reactions, to form the products which are essential for our biological function. Okay. We will further on go to the secondary structure formation. Secondary structure formation of the protein is by the hydrogen bonds, and it is of two types, alpha and the beta sheets. Okay, alpha is the coiled structure in the by the hydrogen bonds, and beta is the plated structure that is formed in the like plates, and alpha is like helix forms. Okay, like our DNA. These globules are formed by the hydrophobic collapse, and it formed the molten globule. The hydrophobic collapse, that is the non-polar part of the amino acids which can not interact favorably with water, aggregate towards the interior part of the protein, and it is so we got the two proteins, alpha helix and sheets. This hydrophobic effect drives its collapse, and this collapse of the protein results in the globular shape of the protein to minimize the hydrophobic repulsions of the polar solvent to the non-polar sides of the chains of the protein, that is amino acids. So tertiary structures, it is the fine tuning of the protein that help to in the hydrophobic interactions, hydrogen bonds, ionic interactions, electrostatic forces between the polar and the non-polar sides of the amino acids. And this lysine and glutamine are the charge sites, and okay, the van der Waals forces are also there, that is a weak and non-specific interaction is present there. Disulfide bonds, that is a covalent bond present between the sulfur. Here we can see that in the I will make you a structure like this. This is the this is the covalent type of bond which is present. It is present in the antibody of our system. Okay, this type of bonds are also formed, and the next structure, it is the quaternary structures, and we see the chaperones. In this, chaperones also help in the formation of the um, protein. Okay, so energy landscape for protein is often described as the unfolding polypeptide, and misfolding or kinetic traps can occur along this pathway, leading to the native globular state of the protein. So the completion of the protein is well defined, and it is protein is a complex structure but flexible, and has hydrophobic and hydrophilic interactions, and core is hydrophobic and hydrophilic is the surface. So to remember that energy should be minimized, and this globule formation is occurs. Okay, bye.